Hello everyone, welcome back to BioBoost. In last video in ER we covered the introduction part, its location, its origin and types of ER. In this video we will continue the same and study its physical structure. So it's a three-dimensional network of intracellular composition and it is formed of three elements, that is cisternae, tubules and vesicles. Moving with cisternae, these are flattened, unbranched, sac-like element. They lie in stacks parallel to one another. And this is the part where ribosomes are present on the surface, because of which RER appears to be rough. It contains two glycoproteins, one is riboforin 1 and second is riboforin 2, which helps to bind ribosomes on its surface. Means these two riboforin 1 and 2 helps the glycoproteins help the ribosomes to bind on cisternae surface. And if in question it is asked what makes the RER rough, then what part of RER makes it rough, then it is the cisternae part where ribosomes are present. Next, tubules. So these are irregular branching element which form a network along with other elements, and these are often free of ribosomes, means these are smooth. Next, moving on with vesicles. So these are oval and rounded, vacuole-like element. These are also free of ribosomes, means both tubules and vesicles are free of ribosomes. All the element of ER freely communicates with one another and contain a fluid called endoplasmic matrix in the ER lumen. Now, one factor which makes RER different is that endoplasmic reticulum may pass from one cell to another through the plasmodesmata in the form of desmotubules. Means it is that organelle of the cell which can move from one cell to another in the form of desmotubules. Now looking at its molecular structure, then the membrane of ER is composed of two layers of phospholipid molecules. Means it is also a bilayer of phospholipid molecule which is sandwiched by two layers of protein molecules like other membrane in the cell wall. Now looking at types of ER, then there are basically two types of endoplasmic reticulum. One is SER and RER. SER is smooth endoplasmic reticulum and rough endoplasmic reticulum. Now we will study SER and what makes it smooth and its functioning and its structure. So basically, smooth endoplasmic reticulum is an arrangement of tubules, vesicles and sacs. Means sacs part is different than the basic structure of endoplasmic reticulum. Means in smooth ER along with tubules and vesicles, sacs are present. The size and structure of the SER varies between the cells. The SER can change within a cell's lifetime to allow the cell to adapt to changes in its function and requirements. We know that there are no ribosomes attached to the membrane surface, and also SER is connected to the nuclear envelope because it is in continuation. Here in this diagram you can see along with nucleus the continuation SER is present in nuclear envelope. The network of SER allows there to be enough surface area for the action of or storage of key enzymes or the products of the enzymes. SER is less stable. The SER is characteristic of cells in which synthesis of non-protein substances take place. Means here